Welcome all. In today's lecture, I will talk about plasmids. I will discuss what plasmids are, what are its different types, and then I will take examples of some of the plasmids, cloning plasmids, I would say, that are currently in use in a molecular biology or a genetic engineering or in a recombinant DNA technology laboratory. So let's start. Before going into a detail about plasmids, I just want to mention that we use one word that is vector. So, what is a vector? Vector is basically a carrier molecule that we use to transfer a foreign DNA into another cell or from one cell into another cell. So, there are a number of vector molecules. There are different types of vector molecules, and plasmid is basically one of the type of the vector molecule. Now, what are plasmids? Plasmids are small, circular, double-stranded, extra-chromosomal DNA molecules, and generally they are found in bacteria. They have the ability to replicate independently inside the host cells, and that ability is due to a region of DNA region which is known as origin of replication site, or we just generally represent that by ORI site. So origin of replication is the region from which replication is initiated, and that's how we have many copies of the plasmid in a bacterial cell. Then these plasmids also have selectable marker genes which are very important. For instance, they have antibiotic resistance gene like ampicillin resistant gene, tetracycline resistant gene, and then enzyme producing gene like Z gene synthesis, like Z gene that synthesize beta galactosidase. So why we use, what is the importance of these selectable marker genes? Now, we know that when we do a transformation of bacterial cells, so when we use a plasmid molecule to transfer our gene of interest into a bacterial cell, as I mentioned in my previous lectures, that we do this transformation, and transformation is one of the inefficient process it's one of the inefficient process. So how to distinguish between those cells, those bacterial cells that have been transformed, and versus those that have not been transformed, are those cells that have taken up our plasmid having our gene of interest, with those bacterial cells that have not taken up the plasmid. So in order to differentiate between these two cells, transformed versus non-transformed, we use these selectable marker genes. So I will discuss later that how we use these selectable marker genes to differentiate between these two different cell types. So one is antibiotic resistant gene, and another one is the enzyme producing genes. Okay, now these plasmids, they range from few thousand base pair to around more than 100 kb. DNA fragment of up to 10 kb can be inserted into a plasmid. So, for instance, if we have a plasmid that is a smaller plasmid, that will have the ability to take, we can insert a bigger gene of interest into that. However, there are certain plasmids that are quite big in size, so then we can insert a smaller DNA fragment into that. However, the total size we can put into a plasmid DNA is about 10 kb. Then these plasmids also carry restriction sites. They are very much essential for insertion of our gene of interest into the plasmid. So that is one of the characteristics that we should have restriction sites, and you know very well that restriction sites are those sites these sites are the regions where restriction enzymes cut the plasmid. So, and these are the sites where we introduced our gene of interest into the plasmid. So, for instance, if we are having EcoR1 site in a plasmid, then we have to cut our gene of interest with EcoR1 site 2, and then we can ligate it into a plasmid. So the idea is that we use similar restriction enzymes both for our gene of interest, for cutting of our gene of interest, and for cutting of the plasmid, is because that would then generate similar sticky ends, which the ligate enzyme will use to ligate our gene of interest into a plasmid. Then the plasmids, they are of different types. For instance, we have fertility plasmid. Fertility plasmids are those plasmids that carry genes, and these genes are essential for conjugal transfer of plasmids. So with the help of these genes, plasmids from one bacterial cells can transfer into another bacterial cell. We have resistance plasmid, as the name indicate. These plasmids, they carry antibiotic resistance genes to make bacterial cells resistant to antibiotics. Yeah, so we have different antibiotics, for instance, ampicillin and tetracycline. So these are different antibiotics. Now, those bacterial cells that carry plasmids with a particular resistance developing gene, antibiotic resistance developing gene in the plasmid, they are resistant to those particular antibiotics. Then we have col plasmid. So col plasmid, they carry gene for colicin, which is one of the bacteriocin, and bacteriocin protein kills other bacteria. For instance, we have col1 gene of E. coli. We have degradative plasmid as well. These plasmids, they carry gene that facilitate host bacterium to metabolize unusual molecules, for instance, toluene and salicylic acid, and we have a gene like TOL gene of Pseudomonas butida, and that's we call them as a degradative plasmid. We also have virulence plasmid. These plasmids, they confer pathogenicity to host bacterium. For instance, we have a tumor inducing a TI plasmid of Agrobacterium tumefaciens that induces crown gall diseases in plants. Okay, we use different types of plasmids in a recombinant DNA technology, and some of the plasmids, they are known as cloning plasmid, and some are known as expression plasmids. Now, cloning plasmids are those plasmids that we use for cloning or making many copies of our gene of interest, while expression plasmids are those that are used to make recombinant protein from our gene of interest. So they are quite different from each other because we have a certain machinery in a expression vector that will be utilized by the host cell in order to make a recombinant protein. We'll talk about that in some other lecture. However, I have just taken example of some of the plasmids. There are so many plasmids, but I have just taken only 3 plasmids that are generally used as a cloning vehicle. So they just make many copies of our gene of interest when we insert our gene of interest in these vectors. So the first plasmid is pSC101, the other one is pBR322, and third one is pUC series vector. This one is like a pSC101 plasmid. As far as the naming is concerned, we name every plasmid based on certain rules. For instance, the small p, it stands for plasmid, and then like SC here in this case, they denote the scientist that created this plasmid, that the plasmid was designed by Stanley Cohen. So that's why we name it as pSC. And 101 is basically any number that is given in a particular laboratory. There is no rule based for these numbering, because in different laboratories plasmids are given different numbers. So pSC101 plasmid, it was designed by Stanley Cohen, and it is one of the, rather it is the earlier known plasmid. So it's a sort of a first generation plasmid. It was designed in early 70s. It is one of the low copy number plasmid. Okay, so what is copy number? Now, copy number means that the number of copies of a given plasmid in a bacterial cell. So when we talk about copy number of the plasmid, that basically means that how many copies of a particular plasmid they are present in a bacterial cell. So some are low copy number plasmids and some are high copy number plasmids. Now the low copy number mean that if the plasmid is present in a very a few copies of the plasmid present in a bacterial cell, then we call them as low copy number. However, if we have many copies of the plasmid, for instance hundreds or 200 or more than that, then we call them as high copy number. So pSC101 is one of the low copy number plasmid, and we use this plasmid for cloning purposes. So if want to make many copies of our gene of interest, we use pSC101 plasmid. The other characteristic or other point is that these plasmids have one antibiotic resistance gene that shows resistivity to tetracycline antibiotic. So this is the overall structure of the pSC101 plasmid. I have mentioned here, so this is origin of replication site. As I mentioned before, that origin of replication site is that region which is used to initiate replication. So and with that replication we have many copies of the of a plasmid. Then it has the rep-A gene. So rep-A gene produces rep-A protein, which is replication initiator protein, and it binds to origin of replication to initiate replication. So rep-A protein basically when it binds to origin of replication site, then the replication is basically started. So once it is started, then it then is taken up by other the process is taken up by other polymerase enzymes to DNA polymerase enzymes to synthesize a complete copy of the plasmid. However, for initiation, this rep-A protein is essential. Then we have restriction sites, as I mentioned before, that these are those regions that are essential for insertion of our gene of interest into these sites. So for instance, by the way, I didn't mention all the restriction sites in the plasmid just to make it more simple, and the actual plasmid will have many restriction enzyme sites. So in this, for instance, I have shown that HIN3, which is one of the restriction enzyme site at position 29. Then we have EcoR1, which is 9262 region, PUV2 7794. So these are the regions. I have mentioned a particular a base pair where this restriction enzyme is located, and then there are certain other restriction enzymes at this region as well. So that is the tetracycline resistant gene. And you can see that there is also a restriction enzyme site, which is BAMH1 at 3676 position. In addition to that, there are other restriction enzymes like SGRA1, SAL1, and PSHA1. And the position of these restriction sites from where they start, they're also mentioned with the restriction enzymes. Okay. Okay, so this one is the next plasmid is pBR322. As far as the naming is concerned, so P is small p is for plasmid, and BR is for Bolivar, and R is for Rodriguez. So these are the scientists who designed this plasmid. And 322, again, like as I mentioned before, that there is no certain rule, no standard rule for these numbers. It's just the number given to a plasmid in a particular laboratory. So the size of this plasmid is 4 kb, and you have seen that in our previous this pSC101 plasmid, the size is 9260, so around 9.2 kb. But this one, the pBR322 plasmid, the size is very small. It is 4.3 kb, so as compared to pSC101. It is also one of the low copy number plasmid, meaning that there are very few copies of this plasmid in a bacterial cell. And to maintain this low copy number, raw protein is involved. I will discuss like what raw protein is. It has two antibiotic resistance gene. So one is tetracycline antibiotic resistant gene, and another one is the ampicillin resistant gene. And ampicillin resistant gene, the gene that give this ability to resist ampicillin antibiotic, that gene is known as BLA gene, that code for beta lactamases, and that confers basically ampicillin resistance, because ampicillin has beta lactam ring. So that ring is broken by beta lactamases, which is a product of BLA gene. And that is basically a BLA gene. Okay, so it is one of the most widely used cloning vector. Now let's come to the structure. So as I mentioned before, the size is 4361 base pair. So here it is origin of replication site. You know very well that this site is used to start initiation and to make many copies. And then this is a rope gene, which stands for repressor of primer, and it serves to maintain low copy number. We have restriction sites at this position. We have some restriction sites at this position as well. As I said earlier, that I didn't mention all these restriction sites just to make it more simple and more understandable. And then we have two antibiotic resistance genes. So one is ampicillin resistant gene, another one is the tetracycline resistant gene. Now ampicillin resistant gene, the gene that give this ability is known as BLA gene, which code for beta lactamases. So you can see that in this region, in ampicillin resistant gene region, there are restriction enzyme sites. So again, we have restriction enzyme sites here as well, like BAMH1, HEN3, and EQA1, and the position from where these restriction enzyme sites start is mentioned in the front. And then we have tetracycline resistant gene. So tetracycline resistant gene has restriction enzyme site for BAMH1 and other restriction sites as well. It has been shown that there are more than 40 restriction sites in this pBR322, and around 10 or more than 10 they are present in this tetracycline resistant gene. So it is very important. I mean, if you compare the structure of pBR322 with the previous, the first generation of vector like pSC101, and pSC101 we just have one antibiotic resistance gene. However, in this case, we have two antibiotic resistance gene. Now, for instance, if we use, any one of these restriction enzyme sites, for instance, if we ligate or insert our gene of interest into BAMH1 restriction enzyme site. So BAMH1 is located inside tetracycline resistant gene. Now, that what will happen? The tetracycline resistant gene will become inactive. So that plasmid, when we incorporate this plasmid into a bacterial cells, these bacterial cells those bacterial cells that have this plasmid, in which we have made this tetracycline resistant gene as inactive by incorporating our gene of interest, they will not be resistant to tetracycline. So they will become sensitive to tetracycline, and they will not grow in a culture media. So that gives us a sort of flexibility that we can use any one of the antibiotic resistance gene for selection. So to distinguish between those cells that have been transformed and that have not been transformed. I will talk about that in few moments. Okay, so the next plasmid is pUC series plasmid. So there are different versions of pUC vector or plasmid, like pUC1, pUC2, and the list goes on, like pUC20, 21, and even more than that. As far as the naming is concerned, so small p is for plasmid, and then UC is for University of California, because they have been designed in University of California. The size is 2.6 kb or 2686 base pair. So when you compare the size of pUC series vector with other vector, for instance, with pSC101, it was very big in size, so it was 9263 base pair. pBR322 is 4361 base pair. So if you compare the size of pUC series vector, they're small vectors. So by the way, this one is this particular vector which I am showing here is pUC18. So pUC18, the size of pUC18 plasmid is 2686 base pair. It is one of the high copy number plasmid. As I mentioned before, that high copy number in plasmid, the high copy number means that these plasmids will have many copies in the bacteria. For instance, more than 100 or more than 200 copies of a plasmid. If copies of the plasmid are present in a bacterial cells, we call them as high copy number. There is no ROP gene. We have seen in our previous pBR322 plasmid that they do have ROP gene, and that ROP gene is required for maintaining low copy number of the plasmid. But here in this pUC series vector, there is no ROP gene. So that's why they are high copy number. There is only one antibiotic resistance gene, which is ampicillin resistant gene, as I mentioned before. The gene BLA gene, it codes for beta-lactamase, and beta-lactamase cleave beta-lactam ring, which is present in many antibiotics. So that's why the bacterial cells, all those bacterial cells having ampicillin resistant gene, they are resistant to those antibiotics that have a beta-lactam ring, for instance ampicillin. Then these vectors also have multiple cloning sites. So multiple cloning site is that region which has restriction sites for many restriction endonucleases, and this is that region where we insert our gene of interest. And there is the selectable marker in this particular plasmid is not antibiotic resistant gene. There is another gene that is like Z gene, which codes for beta galactosidase. So beta galactosidase is used to act on a substrate, so to make a colored product that is that can be easily distinguished when we grow these colonies on a petri plate. So we have a phenomena of like blue and white colonies. So if we do a screening of those cells that have been transformed versus those that have not been transformed, then we get blue and white colonies on a petri plate. So the blue colonies shows that will tell us that these bacterial cells are not transformed, while white colonies will indicate that that particular bacterial cell is transformed. I will discuss that in detail later, but that is quite interesting. And let's come back to the structure of the plasmid. So we have pUC18, the size is that, and we have origin of replication site here, and then ampicillin resistant gene. You can see the different restriction enzyme site in the ampicillin resistant gene. Again, restriction sites are here at this position. Okay, this is quite interesting. Now this you can see that we have a leg promoter region, and this is leg operator region, and that one is, so this region is multiple cloning site, and this is leg Z alpha gene. Okay, now a little bit of more discussion on this particular aspect. So we have a leg promoter. Now this region, this whole region, so leg promoter plus the leg operator, they are like the regulatory region basically. So what happens in that, if we just recall our basic understanding of the leg operon. So leg operon is regulated with the help of operator sequence, which is present in the promoter region. So leg and that sequence of DNA is known as operator sequence. So what happens that the repressor protein, it comes and binds to the operator region. So once it binds to the operator region, then the promoter is not available for transcription. So RNA polymerase enzyme will not bind to the promoter region, and therefore any gene that is ligated downstream to the promoter region will not transcribe. So we have to remove or detach this repressor protein from this operator sequence so that the promoter start works and synthesize our gene of interest. Now in this case, if you can see that we have multiple cloning site right upstream of the leg Z alpha gene. So if we want to insert our gene of interest, we can then put that into this multiple cloning site. But if we do not have any gene of interest inserted into this multiple cloning site, that what will happen, we have to detach the repressor protein, and in that case we use IPTG. So what happens, this IPTG basically attaches itself to the repressor protein, and then it brings some conformational changes in the repressor protein. The repressor protein then detaches itself from the operator region, which is that one, and then RNA polymerase will bind to the promoter region and then start transcribing the gene associated or ligated to this promoter. So in this case, we have leg Z alpha or leg Z alpha gene that is ligated to the promoter. Once that leg Z alpha is synthesized, then we can observe the expression in the form of blue and white colonies, which I will discuss later. So, but the good thing is that these plasmids can also be used as an expression vector because they express our gene of interest. They can express our gene of interest as well. So that's it. Thank you very much.